Look at a pond or a forest. It appears still, but right in front of you, in the water and the air, there is a bustling, living universe that remains completely undetectable to the naked eye. Our eyes have a hard physical limit. When an object is smaller than roughly 100 micrometers, we hit a biological wall where we can no longer distinguish detail. This scale shows exactly what we're missing. A human hair sits right at the edge of our vision. Drop down to 20 to 40 micrometers, and you find pollen grains. Drop all the way down to a single micrometer, and you reach a bacterium. Because we are biologically limited to this 100 micrometer threshold, humanity spent thousands of years unaware of the structures that form the foundations of life. To understand what living things are actually made of, we had to engineer a way to look past our own physical constraints. That solution was the convex lens, a tool designed to bend light and expand the range of human sight. When parallel light rays pass through a convex lens, they bend inward, converging at a focal point to project an enlarged image. But magnification is only half the battle. To actually study a specimen, you need resolution to maintain sharp detail. You can see this physics in action at home. If you fill a round glass flask with water and place it over a book, the curvature of the glass and water acts as a convex lens, enlarging the text on the page. Mastering these optics allowed scientists to take a simple piece of glass and use it to see things that had previously been invisible. In 1665, an English scientist named Robert Hooke used an early multi-lens compound microscope to peer across this threshold. He placed a razor-thin slice of dead cork bark under his lenses to examine its structure. He documented a series of empty, honeycomb-like compartments. Because they reminded him of the austere rooms inhabited by monks, he named these structures cells. By 1674, a Dutch scientist named Antonie van Leeuwenhoek had engineered a radically different device, a single-lens microscope that used a tiny glass bead to achieve high magnification. Using this instrument, Leeuwenhoek became the first human to observe actual living, moving microorganisms swimming freely in a drop of pond water. These discoveries proved that all living things, from a piece of bark to a drop of water, are constructed from individual building blocks. These early observations established that the cell is the unit of life, responsible for every biological process in an organism. In unicellular organisms, a single cell acts as a complete survival machine. In multicellular organisms like humans, trillions of specialized cells collaborate to keep the body functioning. This diagram shows their shared architecture. Every cell is wrapped in a membrane, the gatekeeper controlling what enters and exits. Inside, the jelly-like cytoplasm contains nutrients for biochemical work. At the center is the nucleus, the command center holding genetic instructions. Despite the immense diversity of life on Earth, this shared architecture is the engine behind every organism. While all cells share that core foundation, they undergo structural changes depending on whether they belong to a plant or an animal. Comparing the two, specialized features stand out. Plant cells build a rigid outer cell wall for structural support and contain chloroplasts to trap solar energy. Animal cells lack these, remaining flexible to make mobility possible. By bending light to conquer our biological blindness, we found that the complexity of nature is built from variations of a single microscopic machine.